Welcome to Life and Biology. This is episode 35. We're following up on episode 34 where we talked about cell cycle regulation. In that video, we talked about how growth factors could propel a cell through the first checkpoint of the cell cycle and go on to the S phase where you would copy the DNA. So this slide, I'm using it again. It's the same one I had in the last video, but rather than focusing on the growth factors and how it propels the cell through the cell cycle, we're going to talk about how DNA damage can prevent you from going through the G1 checkpoint of the cell cycle. All right. So in the case of DNA damage, we're going to bring up a famous protein. It doesn't have a great name. It's called p53, which just means it's a protein that's 53 kilodaltons. But anyway, it does have a colloquial name, and that would be it's the guardian of the genome. So how does p53 guard the genome? Well, under normal conditions where you do not have DNA damage, p53 will be made, or with p53 protein is made, it will interact with a protein called MDM. MDM is an E3 ubiquitin ligase, and I will link to a video about the enzyme cascade that leads to ubiquitination in the show notes. But essentially what an E3 ubiquitin ligase does, especially like the ring type, happens to be a ring type of E3 ligase, is that it interacts with E2 enzymes. It's E3, it interacts with E2, so the enzyme before it in the cascade. And E2 would have a ubiquitin stuck to it, and the MDM protein is going to basically act as a bridge to connect to E2 and p53 at the same time, and the net result of that bridge by MDM is that ubiquitin will get transferred over and covalently bound to the target protein. In this case, it's that guardian of the genome, p53. This will happen multiple times. The MDM interacts with multiple E2s that are charged with the ubiquitin, and after those multiple rounds, you end up with p53 that then has a chain of ubiquitins stuck to it. When that happens, especially if it's ubiquitins that are linked to the lysine 48, and we'll talk about that later, but if it's a particular type of linkage of ubiquitins stuck to p53, you'll end up causing p53 to go to this soup can thing. I should have written out the word, but it's a proteasome, proteasome, proteasome. And I always think it looks like a soup can. The main part of the can is loaded with proteases. Those are enzymes that break down proteins. And the top, the green part, the lid of the soup can, is a regulatory domain that can recognize this ubiquitin chain on p53 and cause p53 to be fed into the soup can. And now I'm going to change my analogy from a soup can to a paper shredder. The p53 gets shredded up by those proteases that are part of this here soup can, and out the bottom you get broken down pieces of p53 coming out. So that's degraded p53. Proteins are polymers. When we were learning about monomers versus oligomers versus polymers, we would say that if you have a few amino acids hooked together, you would have an oligomer of amino acids, and that was the word for that was peptide. So when proteins are broken down, you create peptides, and then those will go off and be further broken down by the cell, and those amino acids could be used again for some other protein. All right, but we want to focus on this slide, which is the p53 getting into the action when it acts as the guardian of the genome. Okay, so here's MDM interacting with p53 again. However, there was DNA damage in the cell. You've got a break to both strands of the DNA all this DNA repair machinery comes in and starts trying to fix the break. While it's doing that, you have a protein called ATM, and there's a few others, but I'm making it simple, will go out and put a phosphate group on p53. So last slide we were talking about putting ubiquitins on p53. This time we're putting a phosphate group on. That would cause MDM to be released from p53, and p53 would be able to go, and p53 is a transcription factor, and the phosphorylated form of the transcription factor would bind to the promoter, that's the DNA sequence in front of the p21 gene. That will turn on transcription of p21. You go through the central dogma from reading this DNA to making an RNA, which I'm not showing, to making the p21 protein. And p21 is a protein that can block cyclin-dependent kinases with cyclin, those complexes, the cyclin-dependent kinase complexes. If you remember from episode 34, the CDK and cyclin work together to put phosphate groups on a protein called retinoblastoma, or RB. And if they're getting inhibited, you end up with the version of RB that does not have many phosphate groups stuck to it, in which case the hypophosphorylated form goes and binds to E2F. Now E2F is a transcription factor that was going to turn on the transcription of a number of S-phase genes, but when RB has a low level of phosphorylation, it binds to E2F, and then we've got our red X here reminding us that this complex sitting on the promoter does not transcribe the S-phase genes. So took a while to get there, but DNA damage led to p53 being phosphorylated, skipping a couple steps. p53 turns on a gene called p21. p21 blocks a kinase complex that allows the RB protein to be in the hypophosphorylated form, and it stops E2F from turning on the transcription of S-phase genes. So another action-packed video with lots of details, but trust me, I'm leaving out quite a few more. But I just wanted to give you a good contrast between the previous video in episode 34, where we propelled the cell to go through the cell cycle. The growth factors helped make that happen. And in this case, the DNA damage blocks what the growth factors ended up creating, that cyclin and cyclin-dependent kinase. And so then we end up with these S-phase genes being turned off. All right, well, that was a lot, but that's all I'm going to ask you to know about. I'm talking to my students right now. Anyway, like and subscribe or not. Have a good day.